Thanks for the kind introduction. Can you hear me well?
Yes, very well.
Okay. So I'm okay. It's great honor to be here. Thanks for having us. And actually, we came to visit Araya here. So here, you don't see, but there are offline the audience here as well. So given that we often watch the CC Tokyo, so it's great honor actually to be here. So let me start with introducing the center and LMPO group that we are from. So actually, Professor Hong and I met in the Center for Neuroscience Imaging Research. I mean, that was actually we met before, but before joining here together, but we are at the same center. And this center is actually the imaging center. And so we are neuroimagers, actually. We are using fMRI for our study. Our center has 4 MRI machines, like 2 for rodents and 2 for human and non-human primates. Actually, in our building, there are monkeys and macaque monkeys, and so we have a really nice setup for cross-species research with the functional neuroimaging. And our center director, I didn't know. I learned that he was a committee member of thesis. Thesis committee member for Hiro, who introduced us. Anyway, so. But we share, while we are working on neuroimaging data, we also share deep interest in general artificial intelligence. And so we co-founded this research group called Life-Inspired Neural Networks for Prediction and Optimization Research Group, LMPO. I mean, it's not a big group, but it's we kind of joint lab kind of concept, but we are kind of study together to expand our research interest to AI. And given that my background is in studying pain and emotions and developing fMRI biomarkers of pain and emotions, like basically brain decoding model for pain and emotions. And Professor Hong worked on the clinical neuroimaging and global functional brain architectures and organization. Based on my previous research interest, I'm kind of working on pragmatic domain of our AI. That is like the value learning or reinforcement learning part. And Professor Hong is working on epistemic domain of artificial intelligence, representational learning or world modeling. So we are kind of having a complementary interest, and by doing that, we are kind of integrating new species in this cross-species research program, like mainly the artificial agents. So kind of we are developing our interest. But so today, given that it's been a short time to founded this group, we don't have yet a publication or seeable output, but today we are going to introduce our research direction and interest for future studies and what we are working on, just broad ideas based on some findings from our previous research. So I want to start with this fundamental question: what is pain? Why I'm asking about pain is, mainly just that is my main research topic, but also pain is quite fundamental. Some people say pain is at the pinnacle of consciousness. I tried to find the author, but it's unknown. But it's really true that, for example, anesthesia, that we use that to reduce pain, but that also induced results in the loss of consciousness. And I like the famous Antonio Damasio, who said in this book, feelings of pain and feelings of pleasure, they are the motivation for the intelligence. And also the father of AI, Turing, I think this is a Turing room actually, right? And Turing talked about pleasure and pain systems in his really early work. So pain can be seen as a motivation for intelligence. I'm gonna convince you that that is really the case by looking at the definition of pain. So how people have been defining pain. I mean, there was a revised revision of the definition of pain. So, for example, the top one is the old definition, and bottom one is the recent revision of the definition. But it's kind of similar. But there is a core shared like common theme that is about the actual or potential tissue damage. I'm gonna read this. Pain is defined as an unpleasant sensory and emotional experience associated with or resembling that associated with actual or potential tissue damage. Why this tissue damage is really important, and that body tissue really defines the boundary between me and not me. If you see the body, I mean the skin is the boundary we can easily think about, and that is basically the tissue. And pain is all about the tissue damage. And even for the single cell, I mean, the cell wall defines the boundary between between the organism and the environment. And this boundary is quite important and essential to survive. I mean, for an organism to survive, the existence of boundary is really essential, and that really make a demarcation between the internal and external. And the boundary enables maintaining homeostasis, which is the key characteristic of life. And an organism should have an open boundary. That is like we are for us to become an open system. We need an open boundary to have a close and wise relationship with the external world. For example, for energy harvesting. So the importance of having a boundary also underlies the importance of the internal environment or internal milieu. And all these are actually, I put the quote mark because to kind of say that these are actually coined, I mean, these terms, these are the quote from Claude Bernard, who is the founder of modern experimental physiology. And he coined the term internal milieu and internal environment, and emphasize the importance of this internal environment for the life. And there are some quotes from this book, The Phenomena of Life, common to animals and plants. And the core of these quotes are we need an internal environment to maintain the uniformity here, maintain the uniformity of the conditions of life in the internal environment. And then to have a wise relationship with the external environment, we need that internal environment, and also the stability of the internal environment is the condition for the free and independent life. This is like the autonomous and for the adaptable agents, we need the internal environment. So this really emphasizes the importance of internal environment, and has an important implication for the brain, to the understanding of the brain as well. So there is a well-known, I mean, not well-known, but I want to suggest that there is an internal external axis in the brain. Actually, this is not a new idea. From this seminal paper, the titled "From Sensation to Cognition" by Mesulam, this is the first figure of the paper, and put the motor and sensory system at the left end, and the right end there is a limbic and paralimbic system, and he put the extrapersonal space in this areas, and then internal milieu in this right side of the brain areas. And all these intermediary processing is kind of about the association and heteromodal regions. And so from this paper, he suggests that this internal external axis is really important organizational rule of the brain. And I think it's really kind of again, people recently started to look at the resting state brain fMRI data, and then apply the dimensionality reduction methods on the resting state connectivity data, and then they found the principal gradient, the one main gradient of this resting state fMRI pattern, activity patterns, really follow this external to internal kind of axis. So I want to put the principal gradient really follow this idea. And actually they put this figure in the paper, modified from Meslam, that really kind of consistent with this external internal idea. And also more recently, this Rout et al. Paper, Science Advances paper, argued that there is a global, the global brain dynamics or waves can be understood as a kind of going back and forth between the external versus internal brain systems that are synchronized with arousal. And he had this slide in his talk that when we are aroused, our pupil diameters are enlarged. And that is actually, that has some computational meaning that we are basically doing the epistemic foraging. So we are gathering the information from outside of the world. And that is about the external world and the sensory and visual systems that are correlated with those arousal. And while the default mode and frontal parietal network, they are activated, the pupil diameter gets smaller. And that is more about the kind of managing the internal environment of the body. So it seems that this internal external axis of the brain is kind of reiterated in the recent studies using fMRI or other types of neuroimaging data. So I'm kind of suggesting that this internal external axis is going to be a really important consideration when we want to understand the kind of functional kind of brain architecture, the global brain architecture of the brain. Yeah. And this is not like well confirmed theory or something, but I think this is providing an interesting and important perspective to the understanding the brain. And so I started from pain and boundary and internal external axis, but I want to ask the really important and fundamental question again. And what is really internal? So this is really important question, but it's really difficult to answer. It's easy to think about the internal environment and then external environment and then epistemic foraging and pragmatic kind of motivation to maintain the internal body. But it's not easy to define what's internal. If you think about that, I think we actually in the LMPO group, we have been struggling with this question for a while now. And then I want to provide some hints from my previous work and data. So I want to show first. I want to show some findings. I want to re-examine the previous findings from the paper that I published previously. That is, I want to start with the brain responses to sustained pain. So during my PhD, I usually use the hip pain stimuli so I give the hip pain to people. So that induces like 10 or 15 seconds of hip induced pain. But many of the pain researchers, pain neuroimagers or pain experimenters, they know that is not really generalizable to clinical pain, because clinical pain has a nature of the sustained pain. So sustained of like the long duration of pain is really important characteristic of the clinical pain. And to make a biomarker and to make a predictive model of the brain that is generalizable to clinical pain, we started to study sustained pain, and we started to use the capsaicin as a stimuli. So basically, we apply the capsaicin on the filter paper. That the experimental procedure is quite simple. we apply the capsaicin on the filter paper and then put that on the tongue of the participants. And then we ask them, participants, to breathe only through the mouth, that dry out the liquid. And then the capsaicin stays on the tongue quite kind of stably. And then when they close their mouth, because of the hypersensitivity induced by capsaicin, that really induced pain for a while. Like, this is the rating from the participants. So you can see the red line shows the pain ratings increased by the capsaicin application, and then that goes until like 20 minutes. And during that time, we are getting the functional connectivity, like fMRI data, and then we can calculate the functional connectivity, and we can model the pain. We can model the predictive models to model the pain ratings. And this is there are a bunch of findings in this paper, the 2021 paper, but I want to just show you just one figure from that. This is the functional connections important for, wait a second, this is, important for pain prediction in sustained pain. And here the dots are the regions. Each dot indicate one region. And the color of the dot is the largest scale brain networks. Maybe some of you are not familiar with these networks, but here the sky blue color is the frontoparietal network that is known to be important for top-down cognitive regulation or top-down attention. And here the orange-red color is somatomotor network that is about the bottom-up sensory processing. But here, easily you can find all these lines are connecting different colors, color dots. So that means the important thing is the inter-network connections were really important for explaining this pain, sustained pain intensity. And also as I said, the most important connection was the connection between the somatomotor network and frontoparietal network. As I said, the somatosensory system is bottom-up pain for the bottom-up pain processing, and frontoparietal network is important for top-down cognitive control or top-down attention regulation. And the important thing is these connection is about the connections between the external and internal systems. So the somatomotor network is really for the information gathering about the external world. And the top-down cognitive control and attention is more about the internal goal system and more about internal kind of internal world, internal model. So the connection between regions at different cortical hierarchies are important. That is the second point that I wanted to make. And the next bit is kind of conveying the similar story. This is a follow-up study of the previous slide. We kind of applied another method, a little different method, multi-layer community detection method on the same data, tonic pain data. And then we found that during pain period, pain capsaicin condition, there is a, if you are familiar with the brain structure, you can see the somatomotor system is in this red area. And this red area formed really large community by incorporating many different regions. And this is showing that. So this figure in a purple area is the networks that are appearing in capsaicin condition and also the control condition, no pain condition as well. But the red area is the regions that are incorporated during the pain condition. And as you can see, the red areas include the frontoparietal network regions. As I said, that is a really top-down cognitive control areas, and also basal ganglia and thalamus. And this really, so this forms, what I'm saying is, this during the pain condition, a really large scale network synchronization is happening across the brain systems. And that is consistent with the idea that from a recent study with the phasic pain stimulation, they argue that during pain experience, there is a pain supersystem kind of appeared in the brain. So the point here is that during pain, a large-scale integration occurs across the brain. So what is happening? The point that I wanted to make is actually we can get some hints from the Melzak's body self neuromatrix theory. This is from old work like 1999 paper, and this figure, I love this figure where you might not know Ronald Melzak. He is a big figure in pain research. He is a psychologist, but he made a gate control theory in pain and also more later period he suggested this neurometrics. And here you can see the output of this circle inside of this is a kind of the brain. He think about the neurometrics, and the output here is the pain perception. But interesting thing is the phasing noxious input, the heat pain, for example, heat stimuli is just one of the many concurrent inputs to the brain and to this neurometric system. And there are many different things like, for example, phasic input from the brain, and that is attention, anxiety, and expectation. And also like, if I go here, like there's a like visceral inputs from the inside of the body. And here tonic input from the brain, that is like cultural learning and past experiences. So I want to make the point that even in the context of a hip pain experiment, the pain perception is intrinsically multimodal. So our brain always getting the inputs from many other places, even while we are resting, like we are lying down and rest, our body keeps sending the signal to the brain. And when we are getting for example, sensory inputs from the heat stimuli, we integrate all this information from the outside or inside of the environment to make this pain perception. And that's why I'm thinking the integration across the brain systems is really important for the predicting pain. And it becomes more important too when we want to explain this sustained pain compared to the phasic pain. Because when pain gets stay longer, the inputs from outside, other inputs like other multimodal inputs are becoming more and more important. And I want to just show you another bit of the results from the same ELife paper. That is dynamic changes from the early to late periods pain. And as I said, we apply the same like multi-layer multi-layer community detection methods. And we looked at the community structures, network structures from the early to late period of pain. So early period pain, you feel a lot of pain, and then late period pain, it's getting kind of decreases. And I want you to focus on these two facts. One is somatomotor dominant community, that is a like red color, becomes smaller, while the frontoparietal dominant community, that is a sky blue, that is getting larger, and I will highlight that. So somatomotor dominant community becomes smaller while as time goes on. But as you can see, which brain regions? This is all these regions were in the somatomotor dominant community in the early period, but get separated from that community in the late period. And you can see the DLPFC. If you know the brain structures, maybe some of you are not familiar with that, but these are the frontoparietal network regions. Usually these DLPFC and there are parietal regions. They got separated from they were together in the early period, but they got separated at the late period of pain. And frontoparietal dominant community becomes larger, and these regions show that which regions form the frontoparietal dominant community in the late period. And you can see the, maybe this figure is really good to kind of get the gist of the finding that from the early to late, you can see the frontoparietal network got separated from the somatomotor network and then formed their own community. And the point is, there could be many interpretations. In the paper, we focus on the describing the changes of the network structure. We didn't interpret the results that much. But my underlying hypothesis, potential interpretation could be the sensory discriminative process that is like the processing the external world. Their processes are like really intertwined with the top-down endogenous processes. That is more about the frontoparietal network. So, and this is not a new idea. Actually, I have this idea from the early stages. So 2018, this paper came out in Nature by Harvard Medical School group. They identified, from rodents study, they identified the pathway required for coping behaviors, and they show that that is associated only with sustained pain, not the phasic pain. So when the pain is really short period, lasts really short period of time, the coping behavior doesn't do much. The pathway for coping behavior doesn't do much. But when pain gets longer, to explain that sustained pain, the pathway required for coping behavior becomes much more important. And then to respond, while after reading this paper, I kind of posted this Twitter this post on Twitter. In understanding the difference between acute versus ongoing pain, maybe pain coping seems more important, important than sensation itself. And I think this perspective is really important that, we usually tend to just understand pain as a part of the sensation. But at the core of the pain, sensation is only the part of the experience, and many other things are really important. And I'm saying that internal processes and these multimodal inputs from the body and also many other ongoing activity of the body and the brain, they are all important to explain pain. And the next project data that I want to show you is the My Pain Connectome project. That is still ongoing study. That is inspired by my Connectome project, where the Ross Poldrake, you might know him, that he's a professor at Stanford and big figure in neuroimaging. He scanned his own brain more than 100 times and analyzed the fluctuations of the brain over time. But we scanned, actually, I'm going to show you, we scanned my brain. I get into the scanner many times, but the goal was a little bit different. We did a pain fMRI study to examine the upper ceiling of the accuracy of fMRI-based prediction of pain, and also to disentangle population versus individual level representations of pain. So briefly, I entered the scanner more than 30 times, and I got pain stimuli, and also I got capsaicin in my mouth. This slide is made by my student, and then you might feel that the joy of them. They want to. They are feeling kind of pleasure by giving pain to PI usually. So they happily put my faces in the slide. Anyway, so and we also use the same design for many participants. This is a single scan, but the many participants involve many participants. So the goal was the 30 scans for me, and then 100 people scans for cross-sectional study. And we collected other data. And we finished the data collection a while ago, and then we scanned 33 sessions. And so that's almost 66 hours. And we scanned more than 100 people. And I'm going to call this as a MPC-1E data. Yeah, so my data, I mean, MPC on 1E and MPC 100 data. I want to show you the results. And 5 out of 33 sessions were pain during sleep experiments. So we excluded that from the initial analysis. In pain during sleep experiment, I intentionally slept over the course of the experiment, and I'm getting pain. So I stay overnight, and then I kind of get into the scanner while I'm really feel tired, because I want to know what happens in the sleeping when I get the pain stimuli. Still, whether my brain still respond to pain or not. So anyway, so for further analysis, we use the 28 sessions. So I want to show you the initial preliminary results from this project. So where we aim to predict trial by trial variations in pain ratings. We use the 18 sessions as training set, and 5 sessions for validation, and 5 sessions for independent testing data. And the modeling inputs were, the first input was the population level prediction. So the model trained based on the MPC 100 data, that it was cross-sectional study, so that that model may include the brain representations, brain responses that common to everybody. And the other input was the MPC-1E model that is trained on this, based on these 18 sessions that I call personalized brain model. And the third modeling input was the experimental parameters that, for example, the stimulus intensity or skin size or trial orders that we can think of as an external input, or all these experimental manipulations. And the all results that I'm going to show you is based on this independent testing data set. So no data was in the training procedure. So I'm going to compare the explained variance of these 3 inputs. Remember this color: the blue is the population level, red is personalized brain model, and green is the experimental parameters. So this is the results. The total R-square for this ensemble model of these 3 different inputs were 0.686. That means combining all this information, we were able to explain almost 70% of variations in trial by trial pain ratings. And the population level brain model, the R-square was 0.263, around 25% of variance. I wasn't surprised by this number, because we know that the population level brain model do that kind of level. I saw that number from my previous paper. So in this paper, we tried to explain trial by trial pain rating by combining multiple population level brain models. And then this is kind of a similar number, right? 25.7%. But the interesting thing and surprising to me, what surprised me was the personalized model. R-square was almost 0.60. So we were able to almost 60% of variation trial by trial pain ratings using the personalized brain model. And the experimental parameters, R-square was 0.535, like 53%. And it's not that surprising, because that's the manipulation we are doing. So like giving the high pain stimuli or low pain stimuli. But the point that I want to make is the first point is only with the personalized brain model, we were able to explain about 6% of trial by trial variance in pain ratings. And you can see the personalized brain model can provide prediction above and beyond the experimental parameters, right? So this is the unique variance explained by personalized brain model, almost 12% of variance. So what all this mean? So this is the diagram that I want to show you. So we can say the experimental parameters are external factors that we manipulate. Like we give the high stimulus intensity or low stimulus intensity. So this is external factor that is reflected in the brain. And there is the internal factors that we cannot really explain by the experiments. So you can see the population level models are almost kind of included in the external factor. They are mostly overlap. So we can say the population level brain model explain kind of most of the external factor, but the personalized brain model is cannot be explained by external factors and only explained by the internal factors. And what is the internal factor? That is a really interesting question. What are these internal factors that can be reliably captured only by the personalized brain model? That is a question, and we still need to examine get more data maybe to answer this question. But my interpretation is the individualized and situated context signal that I told you that from this figure, many different inputs from the body or by context or like past experience. All these inputs are internal factors that we cannot easily reduce to the external factors. And the last bit of the results that I want to show you is the study that led to the invitation to this CC Tokyo talk. Hiro saw this paper and he thought this work is interesting, and then he invited me. So I put these results. So it's about spontaneous thought. You might feel that I'm getting more and more interested in these internal factors and what's really internal. And I still kind of not answering exactly, but figuring out what's internal. And then one important thing is the spontaneous thought. We spontaneously just have the stream of thought, as William James said. Then like the William James I said. Anyway, so what is spontaneous thought? There are many different definitions, and you can find the official definition on the web. For example, you can ask ChatGPT the definition of spontaneous thought. But my approach, my kind of conceptualization of the spontaneous thought is spontaneous brain activity patterns from within, like the spontaneous activity pattern. But one consideration is the self-generated thoughts. This spontaneous thought include a strong autobiographical memory component, so influenced by the memory and our past experience. So to study this spontaneous thought, we developed a novel task based on free association, called free association semantic task, where we give the seed words to participants and have them to generate the train of words based on these seed words. So for example, if I give the family as a seed word, this example show that the next word associated with the family was parents, and then the next word was fight, and brother and mother. So the participant has to come up with the free association words every 2.5 seconds, really fast. And then through that, we were able to kind of probe the thought that kind of generated from within. And I want to share, so here, after the word generation period, we show these words, self-generated words, again to the participants and ask them to think about the personal meaning of words. So kind of intentionally, we wanted to induce the kind of episodic memory and autobiographical memory, and people did that. And then, so this is a basic contrast map for the word thinking versus baseline. And as you can see, I showed you that there is an external internal axis, and that is kind of quite aligned well with the principal gradient. That is kind of dimensionality reduction of the resting state brain activity. And you can see this word thinking versus baseline really well aligned with the principal gradient. You can see the default mode network shows activations, and the visual cortex, for example, show deactivation. But it was quite counterintuitive because we actually show the words through the screen, and after I got this map from my student, okay, something is wrong, do the analysis again, and then we got the same map again and again, because I mean the reason why I thought this is wrong is the strong deactivation in visual cortex. And while I know that we show the words to people, and so we decided to apply this finite impulse response. That is about we can model the TR by TR. Every time point we can get the activation estimate, and we use a clustering we apply the clustering analysis on the temporal profile of the brain activity of these regions, and we got 4 clusters. And the temporal profile of the activation is like this. There is a strong visual cortex kind of component that showed a really short transient activation right after the word was shown. So after the participants saw the words, there was a really short time transient brain activity in these visual areas. And then these kind of these yellow and green areas, that is as I said, the default mode network. These regions activation gets stronger after a while. We think that is and we apply the neuroscience decoding. That is we get the terms that has been associated with these regions from the meta-analysis. And this this purple area was kind of the decoding results was visual and sighted. So that is clearly about the visual kind of stimuli, and then we named the stimulus-driven cluster. And these green and yellow regions, the decoding results were autobiographical and memories and remembering and emotional. And these regions show strong activation at the later time. So after I saw this, we started to interpret that as okay, when we got the word stimuli, we respond to that external stimuli really shortly, and then their cognition is directed to the internal world, like the autobiographical and episodic and semantic memory component. So and the other results from this paper was about the post-scan word survey. We got these ratings for the words after the scan. So valence, time, and safety threat, and self-relevance and vividness. So we asked them to rate the word on these dimensions. And just to be a little faster here. So we try to develop the predictive models of these valence and self-relevance ratings. I'm just showing these 2 dimensions because we put that as a main result in this paper. We were able to develop a good working predictive model for self-relevance, but not valence. And we were kind of puzzling because we knew that the valence, we can decode the valence really well. I mean, from the previous paper, like the Chikajo's group, right? The paper showed that valence can be decoded from the brain consistently and reliably, but we failed to predict valence score from this data. And I started to think that maybe the self-relevance play a role in this. And so we decided to divide it and conquer. So we divided the high self-relevance trials and low self-relevance trials and trained the valence model again. And interestingly, we were able to predict valence score only with the low self-relevance trial. And but for the high self-relevance trials, we couldn't predict valence from the brain. And this pattern is showing the predictive model of valence for the low self-relevance trials. And this pattern was able to predict picture-induced negative emotions from the independent test data. So this pattern is generalizable to independent data set. So I believe this pattern really kind of reflect the valence code, but only for the low self-relevance trials. And what happened with the high self-relevance trials? And then this is the kind of main message of the paper. We developed individualized predictive models for valence. And so just one model per person for high self-relevance trials and low self-relevance trials. And both models work quite well. And then, but interestingly, the patterns of the variations of predictive weights across people were way variable for high self-relevance trials compared to the low self-relevance trial. So this showing this variance. I mean the regions that show the variable predictive weights across people are widespread across the brain. The point here is the level of self-relevance was a strong modulator of the brain representations of valence. And I think that really connect, I mean, kind of we can think about this as okay, internal factors really is a strong modulator of the brain representations and changes the brain representations quite strongly. And so I'm kind of trying to convince you that these endogenous top-down processes and also internal contexts are really important. And then previously, in the previous studies, we really focused on extrapersonal space, like external factors. For example, in pain studies, people really want to, I mean, try to correlate the brain activity with external inputs. But new emerging direction is to look at the internal factors and focusing on what is the modulators of these the response to the external world. And just what's the internal again? I mean, I'm asking this question again, and these list of my answers might not be the perfect, but I think the internal is really contextualizing signals from within and key drivers of the dynamic changes in the brain. So even with the same stimuli, we can show the different responses to the external world. And the internal really contains important information about survival because as I said, the signals from within for example, interoception and autobiographical memory, that all include the value information and reward information and self-identity. And so these are all conveying the important information about survival. And from this paper by Andrew Barto and so on, the big names in RL, they ask this question: where do rewards come from? And they say all rewards are internal. And I think that this is really important. It provides a really important perspective to us as well. And I like the Henry Bergson, who is a philosopher, who argued that our everything keeps changing. So here the truth is that we change without ceasing, and that the state itself is nothing but change. So why our brain keeps changing, and it's really hard to capture the gist of the consistent patterns of brain activity representations of something. That's because I think because of the internal factors. So this is my the end of my part, and I think, but just to give the floor to Professor Hong, he's gonna talk about the more nuanced model of the internal, because I kind of explained the internal external axis in a really simple term as a like principal gradient, but it's not only one. And then we need to keep trying to understand this internal external axis. I mean, understand that in a more nuanced kind of way. And
Sure.
Professor Hong will explain that.
So yeah, thank you for a wonderful presentation, Manny. So I'm Seok-Jun. This is my face. Thank you for the invitation, and I'm actually not invited. I just follow Manny and then have a chance to give a talk here. But then what is good about it is my presentation really short. I have only 8 slides, so I'm gonna quickly finish this. So let me start. So there are many wonderful slides from your Manny's talk, but then these are 2 slides that drew my most attention. First one is existence of this external and internal processing axis, which I believe is fundamental brain architecture. And what is the ultimate functional role of this axis is along this axis, the brain gradually embeds moment-to-moment sensory signal into some context to personal situations. So that I completely agree with Wani that this contextualized signals is one of the key component in our brain. So that internal contextualization is one of the most high-level intelligence component, which make each of us here and over the world identifiable and important. But then whenever we present this kind of topic to our center audience, then there is particular question that we've got many times from audience, which is which brain is at the top hierarchy. I'm not sure whether you have asked yourself this question, but this question is also can be framed as another one: which brain area is most internal? Wani already give a definition of his own thought on the internal process. But then at the beginning, I usually just bluntly answer, you see this Maslow diagram, there's nice gradation starting from sensory to the most inner circle, which is heteromodal, paralimbic, and limbic areas. So that probably these are the areas that are most internal areas in your brain. And then people are getting harsher and harsher, and please be more specific, which brain area is really, really most internal. And then I was feeling, a little bit uncomfortable because I think I don't know the answer. And then I keep thinking over and over, why I don't have this answer is probably because this question depends on the perspective, what you are most caring about, internal and external axis, which means that depending on the perspective you have, the brain may have multiple internal states. For instance, if you're caring about this self issue, myself versus non-myself, then as this Daniel's figure shows, but probably the default mode network is the one that you want to first see, because this is a system highly involved in episodic memory and personal experience, which I think highly related to this internal contextualization. And to me, I put the 2 color here because there is internal part and then contextual part in these wordings. But then this DMN is more internal part. And if you now think about what is the structure involved in contextualization process, then then you want to see also hippocampal, entorhinal cortex, and prefrontal cortex, because there are many neuroscientific evidence nowadays to say that this axis actually performs some computational process for the contextualization. And then based on whatever contextual knowledge, your brain is now able to perform state prediction for the futures. So that, in other words, the other name of this axis is generative model. So that by the fact that your generative model always have to be maximally decoupled from your external process area, this generative model can be also internal source. By the way, this is the figure usually people think about whenever we talk about a functional gradient. This generative model, this hippocampal, entorhinal, prefrontal cortex also have this functional gradient starting from hippocampus. Even within hippocampus, the posterior part is most spatially detailed information. And if you come to the more anterior and ventral area, it's more abstract reasoning and cognitive map kind of information. And then you go to the orbitofrontal cortex and BMP, it's more reward and value system, more and more abstract information up to premotor cortex. So you have this spatial detail information up to abstract space to also motor controls areas. So that this is yet another functional gradient you can see from the brain. And then yet another one that you might not want to ignore is this extroception versus interoception perspectives. And then Wani keep talking about this importance of considering these interoceptive signals. And then as this figure shows, this anterior cingulate cortex, amygdala, and insular cortex is the area processing this visceral motor and interoceptive signal, which we collectively call this a salience network. So that as I list here, if you agree with my perspective, then I usually put the self in the center, and then dichotomize two different directions. One is epistemic axis, and the other one is pragmatic axis. And then each of these sections could be really internal source in our brain because they have their own external world counterpart. So that now, whenever I got this question, either which brain is most at top hierarchy or which brain is most internal, then it really depends on your perspective what you're carrying among these three systems. The next question hitting in my mind after having this conceptualization was how the brain actually get to form these three different axes. So that based on my previous research focus, which is brain development, I try to answer this question, which functional network develop more predominantly at first. So that before giving actual finding, I'm going to try to give you a little bit of background. So more specifically, cortical specialization. There are two seminal theory related to cortical specialization. The one is protomap hypothesis, which basically tell you in this developing frontal lobe, you have all differently distributed genetic molecular signal patterns, so that combination of different gradation of these molecular patterning signals, your cortex will start to have specialized areas. So that this is more nature part, and now you come to the more nurture part, then there is also a theory called the protocortex, which emphasizes the functional role of the thalamus. So that as you see in this embryonic brain, along this gestational week, you see this gradually penetrate thalamic axons in the cortical plate. And then this cortical plate serve as a scaffold for the neurons axons, and then these neuron axons start to crawl, follow this red line, and then they finally reach it to the destination in other cortex. So that this thalamic axons is really important external factor for these cortical specializations. And then in this study, I'm gonna highlight the effect of this thalamus. This is the work from my postdoc, Choong-Wan. She has been applied this technique called the connectopic mapping, which I'm not gonna go to details, but give only gist. Basically, if you have an ultra high dimensional brain connectome data, you perform this dimensional reduction, which give you this multiple principal component, 1, 2, 3, and so on. And then this dimension reduced component basically provide you much more interpretable connectome profiles. So for instance, if you want to check the thalamus connectopic mapping, then you want to check the PC1 or PC2. And then there's also counterpart of neocortical projection, how this neocortex is linked to the thalamus. And then there could be multiple components. And then this is example. This shows the thalamic functional gradient, functional organizations, so that it has this smoothly transit connectome profile from anterior to posterior. And then you can also see the counterpart in the neocortex, so that basically you can interpret this red area has a strong connection to the red areas in neocortical projection map. So I'm gonna show you the actual finding in the next slides. So you could also have the different principal component. So that she applied this technique to two different data sets, developing HCP and so HCP is basically Human Connectome Project, and then HCP development. They have really similar name, but then their age target is really different. Developing HCP's target more infant brain, whereas HCP development is child and adolescents. And today I'm going to show you only this finding, which has age range of 8 to 22. So the first thalamic connectopic mapping has this anterior to posterior gradient, whereas in the thalamic connectome, second thalamic connectome mapping is from the top to the bottom, but this actually makes the diverging pattern. So you can understand thalamus has two different large-scale organization. One is from anterior to posterior, and the other one is this diverging pattern from top to bottom. And then now you see this counterpart of neocortical projection. Then you see this red areas has a strong connection to this red part in the neocortex, and the blue areas has strong connection to the other blue areas. And then this is second neocortical projection map, and you can interpret in the same way. But now I try to give really meaningful, the meaning of this neocortical projection map, which I'm not sure whether you know this functional community map. But basically, our brain has 7 different communities over the whole brain. 3 networks consist of external processing areas. This pink area is internal default mode network areas, and then in between there is salience, ventral attention network. So then now if you see which brain area is most red, then you can realize that is this pink, the salience network occupy these red areas. And also in second neocortical projection map as well, as if you see, this is also pink areas. So that our first interpretation of these two cortical neocortical projection map is that thalamus first make the anchor to the salience in the developing brain. And then now if you check the other side of the projection map, then this is the like high order visual cortex, which is external areas. And then in the second projection map, default mode network is the blue area. So that once this thalamus make the anchor to the salience network, then the external axis become emerging from salience, and then internal process also emerging. So that the salience play a role, central role to develop this internal and external axis, which is collaborated in this age-dependent changes also. So that you see, depending on which age you target, this external and internal axis development is much getting more preeminent along the age. So this is my last slide. So in this analysis, I demonstrate, although there are 3 different axes that we can think as most internal areas, it seems that the part that correspond to pragmatic processing areas develop at first, and then probably this DMN and hippocampal entorhinal cortex PFC develop in a later time. So I'm not sure now you have more solid concept about internal model. I'm not sure whether you are convinced now from our talks. As Wani emphasized in his talk, contextual signals from within is such important concept and key drivers of the dynamic state changes, and also contains important information about survivor. Then my finding demonstrated brain development shows manifestation principle of internal and external axis. And that's it. And then we are in CNIR and LMPO group, and then this is Wani's lab, and this is my labs. And then yeah, hope that the talk was interesting. Thank you.